First, thank you very much for having me here. It was a bit short notice for me, and so I prepared my slides very recently, and oh, I didn't know what for a hard act I have to follow now. So that was not nice. So my talk will be definitely not as entertaining. So now, after I worked out how that works, okay, good. So just to give you a bit of background, so as mentioned, I work for the European Molecular Biology Laboratory, and it's a so-called intergovernmental research organization. It's Europe's only one in the life sciences. Many of you will be familiar with CERN for physics, and the European Space Agency and the European Southern Observatory. That are other intergovernmental research organizations which are set up in the same way. And our goal is to do in the life sciences excellent research, drive scientific services for the community, provide advanced training capabilities, but also innovate and translate that into the private sector, and to help integrate life sciences. So we are now 50 years old, and we have grown to now 29 member states. One associate member states, and Bulgaria and Serbia have lined up to also join EMBL. We started with one site in Heidelberg 50 years ago. That's still existing, and it's still our headquarters, where a lot of different areas of life sciences are dealt with. Very early on, two sites were opened in Hamburg and in Grenoble next to synchrotrons, so that we could use there the radiation for structural biology to understand structures of proteins. At EBI, the EBI, where I'm one of the directors, was founded in 1994 and in Hingston, next to the Sanger Institute, which was one of the big sites for, and is still one of the big sites for sequencing various genomes. And the last acquirements were then in Rome, here in Monte Rotondo, which works on epigenetics and neurobiology, exists since 25 years, and Barcelona was founded 5 years ago, working on tissue biology and disease modeling. So we are still working on 5-year plans. So at EMBL, so maybe we are the last communist stronghold in the scientific world. And in these 5-year plans, we say, where do we want to work on? What's our plan for the next 5 years? Because one of the interesting things of EMBL is that we have a so-called 9-year rule. So we don't keep people longer than 9 years normally. So there are only a few foresights like me. Out of the 2,000 people at EMBL, we have only 100 who stayed longer than 9 years. And that allows us to have a quick turnover and adapt to what's going on in the world, and think about what we want to do. And this is fitting very well to the science for sustainability and well-being into the Anthropocene, because we wanted to build a program from molecules to ecosystems. And we want to use and build on our deep rooting in basic science, in fundamental science, to look then how we can make use of that to also tackle societal challenges in human and planetary health. It's not that we want to move to applied science, but how can we bring fundamental science quicker to societal change? So that's where we want to have a bold scientific vision, address global challenges, including antimicrobial resistance, biodiversity loss, climate change, pollution, and emergent pathogens, very much fitting to the topic of this meeting here. And we always bridged across disciplines and sectors, and that's we wanted to bring in also in this field. I'll give you a second to take the picture. Okay, so it's bringing our core areas into new areas. And to do that, we are building so-called transversal themes, which establish and propel then these new research areas inside the EMBL program. Does it go. So these transversal themes are cross-EMBL structures, and it's planetary biology, human ecosystems, infection biology, microbial ecosystems theory, and across all of that, we also have a data science center, which I lead together with my colleague Jan Korbel in Heidelberg, to ensure the coordination of all the biodata activities across EMBL. So I will not go through all of these themes, but I will only touch on these ones, which I think are relevant for what we are discussing here in this conference. And one of the big things is what we worked on is now the planetary biology transversal theme, where we want to understand the interplay of genes and environment in the Earth's biomass and improve planetary health. And our biggest endeavor there is the so-called TREK expedition. Takashi already mentioned the Tara expedition, and what we do now is to go along the European coastlines with a mobile lab, which you can see there on the third picture, parked in front of this white house there in the harbor, and take samples everywhere, do on-site molecular analysis for a start, do a lot of public engagement, and we do that all in collaboration, usually with marine research institutes in our member states. So it's also that we're not only having our 6 sites in 5 countries, but that we can really engage with all our member states everywhere. And while this truck goes along the coastlines on the roads, the Tara schooner is going along the coast by sea, and it's doing also sampling. And to certain sites, so, we will come back from time to time to get also longitudinal data. So, but what I really wanted to concentrate on is our activities in the, not in our research activities, but our so-called scientific services. Because our role is not only to do research, because every country can finance excellent research in their country, so they don't need a European lab there. So we need to, what are the activities which we should do, which are across Europe and should be centralized somewhere, because it's too expensive for every country to do it. And there are certain experimental services, like the beamlines at the synchrotrons, where we have an imaging center and core facilities which are used by many, many experimentalists worldwide. But what I think is probably the most impactful is our data resources, which are at EBI. And so the EBI is, as I mentioned, in Cambridge, actually in a little village called Hingston, on the Wellcome Genome Campus, and it's Europe's home for biological data services, research and training. And what we do there is essentially, we can describe it like that. Scientists generate data and they make discoveries, and then they deposit it with us at the time of publication, the data. We archive and share it with our global collaborators and all scientists, because it's probably the biggest site in the world nowadays doing it, but we work with many, many others together worldwide. Takashi led for many years the DNA Data Bank of Japan, so we are very, very long-standing collaborators on this. And then we also massage the data. We classify, enrich, combine, and analyze the data. We distribute both the raw original data and this value-added data in various types of databases back to the users. And then, of course, we hope that scientists design new experiments on the basis of this shared global knowledge, and the whole cycle starts again. So when we moved from Heidelberg to Hingston with 10 people, we had 2 databases, the so-called EMBL nucleotide sequence database and SwissProt. That were our 2 databases. And nowadays, we have around 40 databases which are very heavily used. When I started in 1987 to work on that at EMBL Heidelberg, we had 200 tapes we wrote every data release and sent it to users worldwide. Nowadays, we have more than 100 million requests per day to our website, and the usage is still growing. We get always new user communities which are now using such type of data. So, and that was really, that we had so much experience in that. That is then also sometimes that you have this infrastructure available, allows you to adapt to certain things very quickly. When the COVID crisis started, then in very short time, we were able to build up a COVID-19 data portal, and I coordinated at that time here in Europe the sharing of the collection and sharing of research data around COVID. So we also worked then with all European and countries and beyond. We not only had this central portal where we allowed people to share data, but we had also in the different countries that people did that when the data couldn't leave the country. And it was not just in Europe, also Japan used our software tools to set up a national portal. What I learned also there was that it's not only important that you do this good stuff and people use it, you need also to tell what's going on, and you need to tell it quickly. In such emergency, you need to tell it quickly. Here's a picture where I was invited to present to the German Chancellor and the 16 Prime Ministers of the states to explain the spread of alpha variant in the UK, because Germany had at that time not a very good genomic surveillance system. Britain had an excellent genomic surveillance system. And so we could see there in real time what was happening and could say, oh, this will also happen in not too distant future in country X, Y, and Z. And we used our European context then to say and to make as quickly politicians aware of what's going on. And I'm pretty sure that helped us a lot to avoid even more loss of life. And I'm pretty happy about that, although it was a tough time for all of us worldwide. But what I also learned is global data sharing is still not an issue which is not solved. Eric is sitting here. You know our discussions in Bermuda, in Fort Lauderdale, in Toronto, where we came up with rules which are followed by the omics community very, very well, how we share data, how we share access, how we share benefit. This is not the case in the case of pathogens. It's not the case in the case of biodiversity. There are a lot of controversies, and the German government brought me into the discussions on the pandemic treaty of WHO as a stakeholder, and I understand now way better why there are certain resistance to complete open data sharing in various parts of the world. Some of it rightly so, some of it weaponized, some of it just lack of understanding. But that's something where we all have a duty to make sure that all of this data which is out there is used for the benefit of humankind. And that's something which is, again, very much in the scope of our meeting today. So I will touch in the last slides a little bit on the use of artificial intelligence. And it's amazing. I mean, we started to have a sort of thinking about how to make better use of artificial intelligence a couple of years ago, and then things just exploded, thanks to people like Demis over here. So we believe at EMBL we have quite a unique position to leverage the potential of growing data in the life sciences and make use of AI in there, because we have very strong research also in AI. We have the resources and services which can profit from it. We do a lot of training of the next generation of scientists. And really we work from molecular level to cellular level to organ level and individual level. And we can make the bridge then in a way that we can really leverage the value of AI in all of these areas. So one thing how we do that is, of course, as I mentioned, we have researchers in data science who develop AI algorithms to use high quality data managed by EMBL EBI to train AI systems. And I will come back to that in a second. But the important thing is here that if we want to have AI systems trained, and Demis pointed that out, we need to have high quality training sets. And if we look at the data which is submitted, a lot of that is not high quality. We need to find out which are the high quality data and make that even better, add value to that, and through expert curation and annotation, and that is then the training set of the future, or also potentially the test set of the future. And the other side is that we can use in our production pipelines already AI approaches to make the whole process quicker. And that happens everywhere. I mean, all the programmers, all the curators are now using AI solutions to speed up their work. And I don't want to give another talk on AlphaFold. That's not my goal. But Demis mentioned that we work together on that. And I just wanted to highlight once again the importance of high quality data. So one of the reasons why AlphaFold worked was that there's a database existing called the Protein Data Bank. It was founded in 1971. It was launched at Brookhaven with 7 structures, 7 X-ray structures. And it was the first open access digital molecular data resource in biology. It's still there, still the single global archive for experimentally determined 3D macromolecular structures. It's now run by, in the US by Rutgers and colleagues, by us at EBI, in Osaka in Japan, and there are, in India and China, there are nodes emerging who will be also joining the consortium. So already early on, this open access database allowed really important observations. So there is this famous plot on the right top corner from a paper from Sarah Shofia and Arthur Lesk, where they showed that structure is much more conserved than the sequence. And that is, for us as biologists, that's now completely obvious, yeah? But this was at that time a new observation. And it is the basis for the prediction by homology modeling. And already in 1969, only on a handful of structures, already the first protein was homology modeled by hand. It was alpha-lactalbumin based on the structure of lysozyme. Nowadays, there's a lot of excellent software packages out there. So two elements really enabled the advances in these structure predictions. One was the open data I mentioned before, and it's not only structural data, it's also genome sequencing and annotated protein sequences. The other thing is that there was a framework to assess it, which was John Mould established the theoretical assessment of structure prediction, and that's where, at some stage, AlphaFold came and wiped the floor, yeah? And they became so successful. And I really have to thank then also Demis and DeepMind, because they came to us and said, look, we couldn't have done that without you. So now we want to give something back to the community. And it was an extremely short timeline. From the first discussions to us, launching it together with DeepMind was 3 months. And I think it was the most important data set we delivered to the public since the human genome. And thanks for that. I think that is for me a prime example how public-private partnerships can work, because we can't do everything in the private sector, but we can also, there are certain things which must be done publicly, but also vice versa. So the last thing is, as I said, we want to make use of AI also in other areas. And I want to give just one example. I was the founder and for quite a long time, nearly 20 years, the PI of UniProt. And what we we already, right at the beginning, we solve a problem that we are just a handful of proteins which are really well characterized biochemically and so on. So if you, molecular function is one thing, yeah, so you can easily, relatively easy say, oh, this is a kinase, yeah. But what is this damn kinase doing? In which processes is it involved? In which tissues? In which developmental stage? What are the interactors? Most of the interactions are transient, and so on. So we started very early on to use relatively primitive machine learning approaches to use this information, classify the known proteins into groups using another database I created called Interpro, and say they have the following domain, they have the following families, then extract from this information, from these well-characterized proteins, the shared knowledge, which is equal to all of them, and transfer to the uncharacterized. So that was pretty primitive, and we had a very conservative approach to minimize false predictions. And we always like to work also with external research groups, because they had much better results usually, but they had no idea what scaling issues or what production ready code needs to be, and external groups may not stick to deadlines. If you run a production pipeline, then you need to deal with that in 2 months, the whole process, and you have for this prediction only a window of 5 days. So what we really want to have is that we control through the literature, then the interesting information is done and it gets summarized. And that's, we can do now. There's this free text there, and in UniProt style, it's then put into this structured pieces. But if we go through that as an example, then we still have sometimes a bit of problem. This sentence, for example, is correct, but it's not in the paper. The other thing is it's twice in there. We don't need to add redundant information. And sometimes this citation, it's just not existing. It's a hallucination. So yes, we want to use large language models, for example, to help us with the efficiency of curation, but we need to work and make them that the curators work in pairs with software engineers. The software engineers need to get new skill sets. And this has a tremendous possibility to improve the efficiency of the curation, but also, of course, all of this needs to be labeled that this was done by a machine and not by a human. And with that, I think I gave you a whistle-stop tour through a lot of the things we're doing, and I hope it was still okay, even if it was not as entertaining as Louis' talk. Thank you very much.